It's so good to be back in California at this beautiful setting, and I want to thank the organizers for the invitation to speak today. One of the most difficult parts about our company is our name. We're Aegle Therapeutics, and there's a background to that which I don't have time to talk you through right now, but we are a pioneer in the development of allogeneic MSC-derived extracellular vesicles. We're focused on rare diseases, including epidermolysis bullosa, and we have a proprietary EV harvesting technology that has really been critical for us to get into the clinic with 2 indications. We have a platform technology. We believe there are indications fairly broadly in multiple other therapeutic areas, and we were a spin-out of the University of Miami. Recent milestones included the transfer of manufacturing from a lab at the University of Miami to a CMO, and that transfer is wrapping up right now, and we hope to be in clinical manufacturing in December of this year so we can launch our first clinical trial in the first quarter. In terms of regulatory, both our INDs for EB and for burns have been cleared by the FDA. And we have fast track and rare pediatric disease designation for our EB indication from the FDA. We have been granted a Department of Defense grant to support our burn trial, the phase 1/2A burn trial. And in terms of intellectual property, our core patents, which cover both method and use, were granted all over the world, and we have augmented that with a number of other follow-on patents. Extracellular vesicle therapy has emerged from MSC therapy, and I don't have to tell 99% of the people in this room that MSCs are multipotent cells capable of differentiating into a whole bunch of different lineages. They're important in the orchestration of tissue repair. They promote angiogenesis, reepithelialization, and granulation tissue formation. What is also very exciting about this group of cells is they are immunosuppressive, they are anti-inflammatory and non-immunogenic, and they contribute wound healing without scarring or retraction. So I want to take you through first how Aegle was basically founded. And it was based on a trial that was done at the University of Miami using mesenchymal stem cells as therapy. It was a phase 1/2A trial, investigator-sponsored trial, that was conducted at Jackson Memorial with 10 patients, and they were treated with mesenchymal stem cells. The results were just published, which is why I'm talking about this data. It was just published a couple months ago, and the results were excellent. The patients saw accelerated healing, and in fact, the patients with the larger wounds saw the most accelerated healing, with 2 of the 10 patients having wound closure rates in excess of 26 centimeters squared per day. The patients all experienced repigmentation. They all experienced skin elasticity and hair growth. And none of them had hypertrophic scarring or contractures. The university measured POSIS scores. This is a scar assessment scale where 1 is normal tissue, 10 is the worst situation. And from a patient perspective, the score was 1.6 after a year, and from an observer perspective, the POSIS score was 1.3. All patients had 100% wound closure, and there were no adverse events or evidence of rejection in any of the patients. Sorry, this is dermatology, but a picture is worth 1,000 words. I'm going to show you 2 case studies. This is a patient who accidentally submerged his arm in hot oil. You'll see on your left an A, that is before the treatment. B is after 7 days. 10 days, and the third on the right is after 17 days or 7 days after the second treatment. You can see complete reepithelialization, and in fact, if you look at the wrist, the way the treatment it was administered, there was an adhesion there, and so clearly there was repigmentation did not happen where the adhesion was. Here's another case of a man who sustained a flame burn while pouring an accelerant into a fire. You'll see before the treatment, this would have been a severe second-degree burn. And on the right, you see a year later, complete healing, and in fact, hair growth that was where the, maybe a little bit further than they normally had, that was on the area of the treatment, but completely regenerated tissue there. So our founder was Dr. Badievos. He's a dermatologist and clinical researcher at the University of Miami. Took a look at what the cells that were harvested from, for this trial, and by the time they were delivered to the patient, noted that there were billions and billions of extracellular vesicles, hundreds of billions of extracellular vesicles in the vial that was administered to the patient. So his theory was that the EVs are actually the paracrine wound healing mediators of the cell, and that perhaps there was a notion here to take this idea forward, to harness the power of these MSCs without using the cells. So thus born is AGLE 102. What is, this is our first product. What is it? It's a composite of EVs that are naturally secreted by MSCs. We use these EVs as a delivery mechanism. They're delivering proteins, they're delivering nucleic acids, and a lot of other incredibly valuable cargo. They're conveying the immunomodulatory and anti-inflammatory aspects and signals of MSCs. They contain bioreactive cargo essential for cell communication, and they're non-immunogenic, and this is a topical application just like the MSC application was. They offer some advantages over their parent cells. They're very robust. They survive in the wound longer than cells. They survive in the GI tract, although we're not using it in the GI tract in this indication. But they don't differentiate, they don't migrate, they don't engraft. And 4 or 5 years ago when I first started talking about this, people would say, oh, aren't EVs the waste of the cells, or at least in part the waste of the cells? Well, today, if you look in the literature, you will not see a publication that doesn't say it's evident that the mechanism of action of MSCs is not by engraftment, but attributable to the transfer of their extracellular vesicles to target tissues. And that's exciting. So how do we make AGLE 102? We harvest allogeneic or donor bone marrow. We culture our MSCs, and then we isolate using a proprietary process. We isolate these EVs to make the therapy. We found that it was very important to design a new process because gold standard processes did not work when we moved from the lab into animals. If the process damages or collects any, say, harsh reagents, you can get an inflammation response. So I'll talk about that in a little bit. There are, just so you know, these include not just the tiny exosomes, but also microvesicles and apoptotic bodies. If in any indication or any therapy you want to start at the beginning, we wanted to make sure that the MSC EV stimulated proliferation like their parent cells, and in fact, in normal adult and diabetic wounds we saw, and these were fibroblasts, we saw again dose-dependent, this would be the 3 bars on the right for both of those, we saw a dose-dependent response in terms of proliferation. And we saw something very similar in terms of angiogenesis. And in fact, in the colored picture all the way on the left, you'll see the tubular formation in color. So other aspects of these EVs that I want to share with you, this was a paper published last year. These EVs are filled with proteins. And in fact, in this paper, we looked at the University of Miami and Dr. Badievos' lab looked at 4 different donors. Each donor had about 3,000 to 3,500 proteins, and in total there were 8,000 proteins identified in this group of donors. Only 642 proteins were common to all of the donors. Of these 8,000 proteins, most were associated with transport, no surprise, transcription, and the cell cycle. This might be hard to see, but on the left is a Venn diagram. You'll see those are that represents each of the different donors with 642 common proteins. And in B, you'll see biological function across the bottom and number of proteins across the side. And you'll see that even though only 8% of the proteins were common to each of the donors, there were striking similarities in the number of proteins per biological function. So in the Wnt signaling, you didn't have 5 in one and 300 in another. They were a very close number in terms of the number of proteins per biological function. There's a lot more to talk about this, but let me just summarize by saying EVs contain a multitude of proteins that are essential for modulating cutaneous wound repair. And they include basement membrane proteins like collagen 7, and that's an important one when you think about our indication of epidermolysis bullosa and the dystrophic form where the body can't make collagen 7. Why do we care about basement membrane proteins? Because they provide structural support and a surface for cell attachment. These also include Wnt signaling proteins, which participate in the modulation of angiogenesis and fibrosis. Very interesting, they contain a whole slew of transcriptional regulators, which are key to jump-starting stalled or faulty proteins, for example, collagen 7 production. And they include multiple DNA and RNA polymerases. So with all of that in mind, we filed with the FDA an IND to move forward with the Burn study for Aegle. And our Burn study is mirrored quite a bit off of that MSC trial that I walked you through earlier. We do have an open IND to begin that study. We are in the startup phase of the trial will begin in the first quarter, and this will be funded by a grant from the Department of Defense. We are very, very focused as well, and our lead indication is dystrophic epidermolysis bullosa. This is a rare pediatric skin blistering disease where the patient cannot make any or in cases deformed or inadequate amounts of collagen 7. So why do we care about collagen 7? Collagen 7 is what the body uses to make anchoring fibrils. And anchoring fibrils are critical to holding the epidermis to the dermis. So if you do not have solid anchoring fibrils, with just minor friction, the top skin blisters off. And these children, this is a pediatric disease. It's a monogenetic disease. Where there's a defection in the COL7A1 gene. And these kids, this is the worst disease you never heard of, or butterfly children, you might have heard that as well. But these kids, 3 hours a day to change bandages, and the bandages can cost $10,000 to $80,000 a month to take care of these kids. So why are we excited about the use of AGLE102 in dystrophic epidermolysis bullosa? First, we talked about these EVs contain the protein collagen 7. We don't think of ourselves as a protein therapy company, but in fact, these EVs do contain collagen 7. But more important, these EVs actually, when you look at them, they express for COL7A1. And when you look at these recessive dystrophic EB fibroblasts that cannot make any collagen 7, they express no COL7A1. When we treat them with our EVs, they do in fact express, and you'll see the bars on the right in both of those primer pair examples. Expression is exciting. But where does what we're really looking at is, can this in fact cause the cells, these disease cells, to make their own collagen 7? And the fact, and in this in vitro study, we took RDEB fibroblasts, we treated them with vesicles, and we removed the free-floating vesicles after 3 days. And in fact, on a dose-dependent basis, these recessive dystrophic EB fibroblasts made their own collagen 7. So let me go back. So really a multifaceted approach to this disease where we're not only delivering the protein, the mRNA, we're inducing the cells to make their own collagen 7, but we're also delivering all of the incredible regenerative and immunomodulating aspects of the parent cell, which is, to me, and the anti-inflammatory aspect of the cell. So that trial will begin in 2022 as well. The IND is approved, and we have, as I mentioned, fast track and rare pediatric disease from the FDA. Our pipeline, we talked about dystrophic EB, we talked about burns. We've also got some very exciting early data in graft versus host, but that will have to wait for another presentation. Let me say thank you all for joining me today, and I think I have 45 seconds for a question. Any questions?